Senye, and in my speech today we're going to be talking about Streptococcus agalactiae. Now, Streptococcus agalactiae is more commonly known as Group B strep or GBS, which is what I'll be referring to it as the rest of the speech. And GBS is a bacteria that can colonize a pregnant mother, and typically it doesn't cause any issues for her, except that when the baby is being born, the baby can become infected, and this can cause a very serious infection. In this speech, we'll be talking about the morphology of the bacteria, the transmission and identification, signs and symptoms, as well as the prevention and treatment. Streptococcus agalactiae, or GBS, is a gram-positive bacterium, meaning that the cell wall is very thick, which is very indicative of just how you're able to identify the bacterium. It is also coccus-shaped and it forms chains, so under the microscope, it will look like a long chain of ping pong balls. GBS is also hemolytic, meaning that it destroys red blood cells. The transmission of GBS is unknown, and it is not sexually transmitted either, so however, its natural reservoir is in the lower intestinal tract and the vagina. The way that GBS is identified is in the late third trimester of a woman's pregnancy. A doctor will swab the anus and vagina of the patient and send it to a lab. And that at the lab, the tech will break the swab off and place it into a nutri broth and allow it to incubate for 24 to 48 hours. After incubation, the tech will inoculate a blood agar plate, which is just a petri dish made with blood. And after incubating it again for another 24 to 48 hours, the tech will observe it for beta hemolysis. And the way this will look is that there will be little colonies all over the plate, and surrounding the colonies will be a clear zone. And the clear zone indicates beta hemolysis, which means that that particular colony would be positive for GBS. If there are no zones of beta hemolysis found on the blood agar plate, then that would mean that the patient is negative for GBS. However, further incubation will be done to make sure that this is the case. Now, the way that the baby can be infected is typically when a baby is born prematurely, meaning that the mother has not been tested for GBS yet. Also, laboratories can mix up people's results, or have a false negative result somehow, and the infection goes without notice. Now, when the baby is being born, the bacteria can come in contact with the eyes, nose, and mouth, which is typically how the baby is infected. Another way the baby can be infected is while the baby is still in the uterus and the mother's membranes have been broken for over 18 hours, and meaning that the bacteria travels from the vagina up the cervix and into the uterus, where it'll enter the eyes, nose, and mouth of the baby. Early onset infection occurs within 24 hours to 1 week, and late onset infection will occur between 1 week and 3 months. Now, there are a few signs of infection in both the infant and the mother that can occur after birth. In the infant, the baby can have a fever, have trouble breathing, or The mother can also have an infection that can be very dangerous as well, and she may experience when the placenta detaches from the uterus. There is a large hole, not hole, but wound left in the uterus where the bacteria can infect her. Then the baby may experience sepsis, bacteremia, meningitis, and pneumonia, and all of these infections are deadly. Well, there's no vaccine. The only sort of prevention that can take place is that the mother is tested early enough before birth. Sorry. The only prevention that can take place is just knowing that the bacteria occurs and to administer antibiotics in time. And the way that it is treated is that there are antibiotics that are given to the mother at least 4 hours before birth through her IV. So that was a brief overview of Streptococcus agalactiae, or Group B strep. Group B strep is a typically harmless bacteria that is present in many people. However, it can be very dangerous to the infant. GBS has a specific morphology, and it is very easy to identify in the lab. The infection occurs during birth or during labor, and the infection can be very deadly in the baby because it can cause sepsis, bacteremia, pneumonia, meningitis, and there is no current prevention from the mother being colonized with GBS, but right now we can prevent the infection spreading to the infant by using antibiotics before the baby is born.